Hello everyone. So today I'm going to start a cell division and cell cycle topic. So it's been so many days, so I didn't get time to post this video. So sorry for the inconvenience. Let us go into the cell division and cell cycle. So generally, what is the meaning of cell division? Cell division means one cell is dividing and giving other cell. The old cells will give new cells. The already existing cell will give new cells. So you can see in this image, one cell is getting doubled into two cells. You can also see the chromosomes which are duplicated or replicated and going towards the opposite cells. So this is what cell division looks like. So in the next slide, you can see in detail about cell division. Cell division. Cell divides to give new cells. One cell will produce two new daughter cells after division. So we are all know that. So you can see in the picture, we are having two cells with two chromosomes, two nucleus, so like that. So when will cell division will going to start? So whenever they reach a desirable size, then only they'll start to reproduce. Okay. So this cell division is also called as self-reproduction. Self-reproduction means the existing cell, the old cell will produce two new cells. Next slide, you can see how cell division is regulated. So the regulation is very important for cell division. Without cell regulation, it may lead to cancers. Unregulated growth of cells will lead to cancers. So that is very important. Cell division should be regulated. One cell has to do two cells at a minimum particular time period. So you can see here cell division is regulated and coordinated with both cell growth and DNA replication to produce intact genomes. See here, genome means the DNA, which is very important. The most important molecule in cell is DNA, which is having total information about the cell. So that DNA has to be not damaged. So if it is damaged, the next generation, next new cell will not get the complete genome. So that's why cell division regulation is very important. Very important. So next, what is DNA replication? So it is the process of genetic material get duplicated into two copies of chromosomes. For example, if I take an organism contains two chromosomes, it will produce four chromosomes. These two chromosomes are replicated into four chromosomes. You can see on the right side here, DNA replication, double-stranded DNA. This is a double-stranded DNA in eukaryotes, like humans and plants. So this is template, then this is new DNA. From the template, the new DNA is synthesized. So this is called DNA replication. So two chromosomes are there. The two chromosomes will produce four chromosomes. Means doubling of the chromosomes. So why? Because they are doubling to give it to the next cell, next to new generation cell. If cell division is not properly regulated, many damages to the organism will occur. So that will lead to the cancers. Already I mentioned it. Now next we'll see what is cell cycle. Cell cycle. Cell cycle is the phases cell undergo to produce new cells repeatedly. In cell cycle, we have phases. One phase to the next phase, they will be in a cyclic manner. Like we have G1, S, G2, and M. They will be repeating again and again when there is a cell division process. So this cell cycle, this is called as cell cycle. Cell cycle is divided into two major parts. One is mitosis, and another one is interphase. Further, they are divided into four phases. First of all, we'll see interphase. What is interphase? Interphase contains G1, S, and G2 phases. These three phases together is called as interphase. After interphase, we will enter into mitosis. Mitosis means the duplication of DNA, the separation of chromosomes to the next cells. So when we see G1, G1, S, and G2, what is the meaning of G1? G1 means it is called as gap 1 phase. Gap. What gap here? Here gap means, so it's a cyclic process. G1, S, G2, and next comes M. After M, the cells will enter into G0 phase, and then they will go into G1 phase. So there is a gap between mitosis, M phase, to next to G1. So that gap is called as gap 1 phase. Next, coming to the S phase, S means synthesis phase. Here, the DNA is synthesized, the new DNA from the old existing DNA. The new DNA is synthesized by semi-conservative mode of replication in eukaryotic organisms. And next, coming to the next phase, that is G2, it is called as gap 2. Second gap phase. After synthesis of DNA, to the next entering into the G2, that period is called as, that phase is called as gap 2 phase. Now we'll see in detail about this individual interphase G1, S, and G2, and later we'll see what is mitosis. So coming to cell cycle phases, how much time they will take during the cell cycle? Cell cycle time for cell cycle. Cell cycle duration is varied in different species of cells. The different species will be having different time period. For example, here I'm taking example of human cell, which is dividing in a laboratory conditions. It will take 24 hours approximately to divide one cell into two new daughter cells. So it will be taking 24 hours. So when we see the time taken for different phases, so here you can see G1 gap 1 phase will take approximately 11 hours to complete. Next S phase, it is also called as synthesis phase of DNA replication phase. It is having 8 hours. Next G2 phase, it will take 4 hours. M phase, that is mitosis, it will take 1 hour. You can see the variation here. Almost all G1, S, and G2 is taking so much time. Hence, the interphase is very, very important and mostly time-taking process. Like approximately it is 22 hours. 22 hours. It is in the interphase. Only mitosis takes 1 hour. Right. Next, you can see in the right side diagram what happens in G1 S and G2 phase. You can see here in the G1, it's a cell and its nucleus, and it is having DNA. Unreplicated DNA is the G1 phase. Unreplicated DNA is present. So in this gap 1 phase, all the materials, all the proteins, all the enzymes required for the cell division are synthesized here. So that's why it is called gap one phase. It will take some time to produce all these things. Here in this gap one phase, there is no replication of DNA. And when you go to S phase, the DNA is duplicated. It's doubled. That is called replicating DNA. You can see in the picture. For this S phase, G2 phase, these chromosomes are completely duplicated, and you can see two chromosomes, two replicated chromosomes in the G2 phase. Okay. So this is the things happening in G1, S, and G2. Next, we'll move on to next slide. So that is the phase G0 phase. What is this G0 phase? When we are having interphase in mitosis, after cell divides into two cells, then it will enter into no activity, no metabolic activity. So that phase is called as G0 phase or gap zero phase. Cells also have G0 phase, where cells metabolically active, but they will not divide. And they will not synthesize the proteins, and they will not participate in cell division. So it can be seen in the nerve cells, neuron cells. So that phase is also called as quiescent phase. You can see in the picture, these are the neurons. So we know that neurons are the brain cells. They will not divide again and again. The lifetime they will divide only once, and after that they will not divide. If we get damage to our brain cells, they are not replaced. Okay. So some other cells are there. We are having other cells. They can be regenerated in our body, but these brain cells are not regenerated. Why? Because they have entered into not cells. And also we have some other cells like skin cells, skin fibroblast cells. They can be replaced. They will enter into G0 phase, and again they will enter into G1 as G2. So like that, they can be activated to enter into cell division. So this is one of the important phase of G0 in cell cycle. Now I'll see how cell cycle is regulated. How it is regulated. That is called regulation of cell cycle. So this cell cycle is regulated mainly by two factors: external factors as well as internal factors. So these are called as external signals or internal signals. Now you see external environmental signals such as the scientists have observed in Saccharomyces cerevisiae in the yeast. If the cell is not reached a required size, it will not undergo cell division. So